4,000 meters below the surface, the Atlantic abyssal plain exists in a state of perpetual darkness and crushing pressure. This extreme environment is home to the order Rajiformes, specifically the family Rajidae, known as the hard-nosed skates. The family is vast, containing at least 154 described species organized into 17 distinct genera, found in every ocean on Earth. For the taxonomist, these animals present a significant challenge. Their flattened, subrhomboid discs and dark cryptic colors make them appear nearly identical to one another. These similarities result from convergent evolution. Their bodies are optimized for a sedentary life on the sea floor in temperatures that rarely rise above freezing. Identifying a skate's true lineage requires looking past its silhouette. Because the environment demands visual uniformity, we have to isolate the specific micromorphology of the animal. Biologists navigate this complexity using a dichotomous key, a system of binary anatomical choices that filter specimens through a sequence of specific physiological traits. This method of classification is rooted in the history of cartilaginous fish research, much of which was advanced by oceanographers like Henry B. Bigelow. We are going to apply these filters to an unidentified specimen, a dark brown skate recently retrieved from the deep Atlantic. To find our specimen's place on this tree, we will analyze 3 distinct biological systems. This anatomical filtering allows us to accurately map biodiversity in regions of the ocean that remain largely unexplored. The analysis begins with the head, specifically the short, blunt angle of the snout. Inside the snout, the rostral cartilage defines cranial rigidity. Our specimen's central beam is stiff and stout, unlike more pliable genera. Moving to the wings, a distinct semi-translucent area clearly separates the pectoral fin skeleton from the tip of the snout. This skeletal gap allows us to definitively remove genera like Breviraja and Neoraja from our list of possibilities. Internal skeletal anchoring serves as the first absolute filter in skate classification. Next, we move to the skin surface to examine the distribution of dermal denticles, commonly known as thorns. Every species follows a specific map for thorn distribution across its dorsal surface. In our specimen, orbital thorns surround the eyes, with nuchal thorns at the nape. Scapular thorns reinforce the shoulders, and a continuous median row runs to the first dorsal fin. We also look for evidence of sexual dimorphism. Adult males develop specialized, retractable thorn patches. Our specimen possesses both malar and alar thorns, located on the far outer regions of the pectoral fins. These thorn distributions function like a biological barcode, allowing for precise identification even when body proportions are identical to other species. The final division in the key requires an analysis of male reproductive anatomy. We isolate the pelvic region to examine the clasper glands, a complex cartilaginous structure. Our data narrows the search to just 2 genera, Leucoraja and Rigella. Leucoraja is defined by external cartilage components called the promontory and the roll. Our specimen entirely lacks these components, matching the other branch. We cross-reference this with our thorn map. Unlike some related genera, adults in this group retain their continuous median thorn rows throughout their lives. The evolutionary split between these deep-sea lineages is recorded in these millimeter-scale variations in cartilage. With the key resolved, the combination of stiff rostral cartilage and this specific clasper morphology identifies the genus as Rigella. Our specimen is Rigella bigelowi, commonly known as the chocolate skate. Its distribution is disjunct, spanning the mid-Atlantic ridge and Rockall Trough to the Gulf of Mexico. It lives in an extreme niche, recorded at depths exceeding 4,000 meters, in temperatures as low as 3.5 degrees Celsius. This benthic predator uses its flattened body to hunt small crustaceans along the continental slopes. Rigella bigelowi represents a highly engineered biological machine, perfectly adapted to the abyss. The survival of the species depends on a reproductive cycle built for long-term stability in these near-freezing waters. Like all members of its family, the chocolate skate is oviparous, laying eggs in the deep. These egg cases, or mermaid's purses, are encased in a tough layer of keratin, with stiff, pointed horns at each corner. These horns anchor the capsules into the muddy or sandy benthic flats, preventing deep ocean currents from sweeping them away. Development is slow. In the cold of the abyss, it can take up to 2 full years for the embryos to hatch. In the dark, deep ocean, survival relies on the precise perfection of this biological architecture.